Welcome to the deep dive. You ready to dive into some cool new cancer research?
Always. What have you got for us today?
Okay, so we're looking at this new drug, and get this, it targets CDK1213.
CDK1213, interesting.
Yeah, and the coolest part is AI helped discover this target.
Oh, wow. So AI is designing drugs now.
Well, not quite designing the drug itself, but it pointed the researchers in the right direction. They use this platform called Pandomics.
I've heard of that. Isn't it the one that analyzes tons of data, like all the genomic stuff, scientific papers, even clinical trial results?
Exactly, and that's how they landed on CDK1213 as a promising target. And there's a whole detective story here with early lab results, mouse models, even early safety data.
Okay, I'm hooked. Let's break this down. First of all, what exactly are CDK12 and 13? I mean, I know CDKs are involved in cell cycle regulation, but...
All right, but these aren't the typical CDKs that are usually targeted in cancer.
Okay, so what makes them so special?
Well, they regulate DNA repair genes.
Oh, no, that is interesting. So if those genes aren't working properly...
Big problems.
Yeah, you're going to have errors in DNA replication, repair goes haywire.
And that leads to...
tumors, especially the kinds that are resistant to our usual treatments. So that's why hitting these specific CDKs is so exciting, you know? It's like a whole new avenue for attacking those tough cancers.
I love that. And this Pandomics thing, did it just spit out CDK1213 as, like, the magic bullet?
Not exactly. It analyzed all that data, like you said, and ranked potential targets based on certain criteria.
Like what kind of criteria?
Well, things like how druggable is this target? Is this something new and novel? What about safety concerns?
Makes sense. You don't want to chase after a target that's impossible to hit with a drug.
Exactly. And when they looked at everything, CDK12 really stood out. And not just for one type of cancer, but across many different types.
So they had their target. Did they just jump right into designing a drug?
Well, there was an earlier attempt. They called it Compound 1, but it didn't quite work out as planned.
What happened?
Turns out it was a bit too potent.
Too potent. How do you even...
Yeah, it caused some pretty nasty side effects. The researchers had to stop the experiment because the mice they were testing it on started losing a ton of weight. Like, way too much, way too fast.
Oh, yikes. So back to square one.
Not exactly. They learned a lot from that failure. And it's kind of the good example of the balancing act researchers face, right? You want a drug that's effective, but not so strong that it harms the patient.
Absolutely.
Yeah.
So what was it about Compound 1 that caused these problems?
Well, it was what we call a covalent inhibitor.
Okay, I gotta ask, what does that even mean? Covalent. What's the difference between that and a regular drug?
You know how most drugs bind to their target, kind of like a key fitting into a lock?
Yeah.
You put the key in, it does its job, you take it out, it's reversible.
Okay, I'm following.
But a covalent inhibitor is different. It forms a permanent bond with its target. Like, imagine supergluing that key into the lock.
Whoa, okay, so that's why it was so potent. It basically stuck there and wouldn't let go.
Exactly, and that's where the potential for side effects comes in. You need to be really careful when designing a covalent inhibitor because you don't want it to bind to things it's not supposed to.
Makes sense. So that's what they had to figure out with their next attempt, right? How to make a covalent inhibitor that was still potent but also safe.
Exactly. And that's where the story gets really interesting. They designed this new compound, compound 12B, and they made some really clever tweaks to it.
Okay, I definitely want to hear about those tweaks. But first, we got to take a quick break. We'll be right back with more on CDK1213 and the amazing potential of AI in drug discovery.
So before the break, we were talking about how they had to go back to the drawing board after that first compound. You know, the one that was a little too enthusiastic.
Yeah, Compound 1, the superglue key. So what did they do to fix it? How did they come up with Compound 12B?
Well, the first thing they did, obviously, was make sure that 12B was actually hitting its target.
CDK1213, right. But how do you even, see a drug binding to a protein?
There are ways. They used a technique called peptide macking. It's pretty cool, actually. Basically, they can see if the drug is attaching where it's supposed to.
Okay, so they confirmed it was binding, but was it actually doing anything to the cancer cells?
That's the million-dollar question, right?
Yeah.
So they took a whole bunch of different cancer cell lines.
Like what kind of cancer?
Oh, you name it. Leukemia, breast cancer, colon cancer. They even threw in some melanoma. And they exposed these cells to 12B in the lab.
Drum roll, please.
And it was amazing. This stuff was incredibly effective at stopping those cancer cells from growing across the board, pretty much.
No way. So it wasn't just a one-trick pony. It worked on all these different types of cancer.
That's what's so exciting about it. But they didn't just want to see that the cells were dying. They wanted to know why. How is 12B actually working its magic?
So going back to what we were talking about earlier, with CDK1213 being involved in DNA repair.
You got it. Remember, if you block those CDKs, those DNA repair genes they regulate,
They don't get expressed properly, which means...
DNA damage starts to build up, and that's exactly what they saw in the cells treated with 12B. It was like a cascade of events, all leading back to that initial inhibition of CDK1213.
Wait, so they could actually trace those broken DNA bits back to the drug doing its job? That's amazing.
It is, isn't it? It's like a smoking gun. And here's another thing. Even after they removed 12B from the cells, the DNA repair was still messed up.
Oh, wow. So it wasn't just a temporary effect.
Nope. Which, again, points to it being a covalent inhibitor, you know? It's formed that lasting bond, and it keeps working even after it's gone.
Okay, so it's working in a dish, but what about in a living, breathing animal? You can't really model all the complexities of the body in a petri dish.
Right, so they took the next step and tested it in mice. They actually used 2 different mouse models, one for breast cancer and one for leukemia.
Did it work? Did it shrink those tumors?
It did. In both models, compound 12B showed significant anti-tumor activity. It was really quite remarkable.
That's incredible. But hold on. Didn't we say earlier that the first compound, that super potent one?
Compound 1.
Yeah, it caused all those awful side effects, the weight loss and all that. Did they see anything similar with 12B?
That's the best part. They observed no significant weight loss or any other major side effects, even at the doses that were clearly effective against the tumors.
Whoa. So they managed to keep the potency, but ditch the toxicity.
It seems so, and that's really the holy grail of drug development, isn't it?
It is. But, I mean, mice aren't people. What happens next? This is all still preclinical, right?
Right. The next step is, of course, to move into human clinical trials.
Which is a whole other can of worms. But I guess we'll save that discussion for the next part of our deep dive. Okay, so before the break, we were talking about clinical trials and, how that's the next big hurdle for this drug, compound 12B. But you also mentioned this thing about resistance. Like, could the cancer cells figure out a way to outsmart the drug?
Yeah, that's a big concern with any cancer treatment, right? Especially something as targeted as this. It'd be like, those bacteria that become resistant to antibiotics. Except it's cancer cells.
So how do the researchers try to stay ahead of that?
Well, there are a couple of approaches. One is combination therapy. Instead of just using 12B on its own, you might combine it with something else.
Like what kind of something else?
It could be traditional chemo, radiation, or even another targeted therapy that works in a totally different way. The idea is to hit those cancer cells from different angles, so it's harder for them to develop resistance to any one thing.
Makes sense, like overwhelm them with an army instead of just sending in one soldier. But, what if that's not enough?
Well, that's where the search for new drugs comes in. And this is where AI could really start to shine, even more than it already is. Imagine using AI to not just identify targets like CDK1213, but to actually help design the drugs themselves.
Whoa, that's next level stuff. So instead of just sifting through data, it's like creating something totally new.
Exactly. AI algorithms are getting so sophisticated that they can analyze huge libraries of chemical compounds and look for patterns, predict how they might interact with different targets in the body. It's incredible.
Okay, so we're talking about speeding up the drug discovery process. But what about personalized medicine? That idea that we could tailor treatments to each individual patient. Could AI help with that too?
I think that's where it has the biggest potential. Imagine taking a patient's genetic profile, looking at the specific mutations driving their cancer, and even factoring in how they've responded to past treatments. AI could help doctors sort through all of that information and come up with a treatment plan that's truly customized.
Wow, so it's like having, a team of super smart doctors working 24/7 to analyze your specific case and figure out the best approach. That's amazing. And this is all, on the horizon? Possible in the near future.
I think it's closer than we realize. And it's not just limited to cancer either. Think about all the other diseases out there. Alzheimer's, heart disease, infectious diseases. This kind of AI-powered approach could revolutionize how we treat, pretty much everything.
It's pretty mind-blowing when you think about it. We started this whole deep dive talking about one specific drug, and now we're talking about, the future of medicine.
That's the beauty of science, isn't it? It always leads to more questions, more avenues to explore. And this research on compound 12B, it's just one piece of the puzzle. But it's a really important piece because it shows what's possible when we combine scientific curiosity with cutting-edge technology.
Absolutely. And it gives me so much hope for the future. So to everyone listening, thank you for joining us on this incredible journey into the world of cancer research and AI-powered drug discovery. It's been an amazing conversation, and I hope you've learned as much as I have.
And remember, stay curious, to keep asking questions. You never know what amazing discoveries are just around the corner.
Until next time, keep diving deep.